Section 11.3 is about transduction. The main ideas of this section are signal transduction pathways, protein phosphorylation and dephosphorylation, and small molecules and ions as second messenger. The main components are cyclic adenosine monophosphate (CAMP) and calcium ion, an inositol triphosphate (IP3). An enzyme called adenocyclase, which is embedded in the plasma membrane, converts ATP to CAMP in response to an extracellular signal called epinephrine. IP3 is a second messenger that is produced by the cleavage of a certain kind of phospholipid in the plasma membrane. It stimulates the release of calcium from the endoplasmic reticulum.
This cell receptor is activated.
A relay molecule is then activated, which in turn activates the first protein kinase. Protein kinase 1 then transfers a phosphate from ATP to an inactive molecule of protein kinase 2, thus activating the second protein kinase. The second then mimics the first protein kinase, phosphorylating a third protein kinase. The second protein kinase is then dephosphorylated by protein phosphatases, and reduced to its inactive state. Finally, the third protein kinase phosphorylates the protein that brings the cell's response to the signal. The protein kinase 3 is then dephosphorylated by protein phosphatases. The first messenger activates a G protein coupler receptor, which activates a specific G protein. In turn, the G protein activates adenocyclase, which catalyzes the conversion of ATP to CAMP. The CAMP then acts as a second messenger and activates another protein, leading to cellular response. After a G protein is activated and cleaves the IP3, it diffuses through the ER, releasing calcium ions, which activates proteins in cellular response. The most difficult part of our selection is understanding how the second messenger, CAMP, is created, which is through the enzyme adenocyclase. Signal transduction pathways are protein-based processes that at each step the signal transduces into a different form due to change in protein shape by phosphorylation. The process of dephosphorylation returns the activated protein kinases back to their inactive state. This process continues until the specific protein needed carries out the signal, activating the last protein kinase.